So thank you for that introduction. So yes, formally, actually, it's been 3 weeks since I was with the FDA. So I'll have to speak FDA-wise for this part, but for the panel meeting, I think I will do a lot more personal input. So I just wanted to put that up front, that'll be more of a personal view of what I've seen throughout the Ebola pandemic and how adjustments had to be made across the board as different information came in about Ebola as a disease. But let's start off with talking about a general overview of how FDA regulates in vitro diagnostic devices. So these are devices that are tests, basically the tests that you have done when you go to a physician. Then I'll talk about emergency use authorization. I'm not going to duplicate what's been given by my colleague already, although you can see that a lot of it was focused on drugs and vaccines. Diagnostics were a little bit different. We have more flexibility, and trials can be done much quicker for an in vitro diagnostic than for a drug or a vaccine demanding to have patients enrolled, etc., etc. We learned a lot of lessons how to do the emergency use authorization, how to utilize it during the 2009 H1N1 influenza pandemic. What we realize now, and I'll bring that up, is yes, that was an easy one. We knew about influenza, we knew about the pathology of it. It was mostly protecting the U.S. Patients and population. Very different what occurred with Ebola, and some of those lessons we certainly used, but there are a lot of lessons that we've learned from the Ebola outbreak in terms of how we could apply some of those lessons from H1N1, and I'll try and cover that too for you. So again, the FDA's Emerging Threat Mission, you've heard about that from Cindy, and it does include appropriate diagnostic tests. There was a time when people just thought FDA, when they were talking about FDA oversight, just talked about drugs and vaccines. Well, that's changed a lot. And now when people talk about FDA's oversight, they do include diagnostics. We do have to assure the safety and effectiveness of regulated medical products in a similar way as the drugs and the vaccines. And we have been involved with the CBRN, emergency preparedness and response capabilities, which, as you'll see, actually made a lot of the different agencies across the board in HHS and DoD start working together. If you'd have talked to people 10 years ago, you would have found that CDC were following this track, DoD were following that track. There was even duplication. And somebody said, the government has only so much funding. We really should start working together. And so some of these emerging threats actually have brought all of the different agencies to start working together and have meetings. So that's one of the pluses that came out of some of these emergencies. You've heard about the review balance. The main objective, of course, is public health needs. What I mentioned just now was until 10 years ago, the focus of the FDA was the U.S. public and U.S. public health. That's changed dramatically over the last 10 years, not just in terms of trying to prevent an epidemic from morphing over to the U.S., which is the case with Ebola and MERS-CoV, but also the kinds of drugs, devices, and maybe not vaccines, but diagnostics being produced outside of the U.S. and being brought into the United States. That's more of a compliance issue. That's trying to regulate and make sure that what's available to the public on the internet, etc., has actually been looked at and carefully reviewed to look at safety and effectiveness. So the public health needs used to be for the FDA, the US. Now it's more of a global mission. There are the regulatory requirements. There are laws that have to be looked at, flexibility applied where appropriate, and above all, the science. The bottom line that I always tell manufacturers is it's your scientists, we're scientists. The science is the bottom line of any approval of any of the medical products that are looked at. So FDA's mission in terms of Vitro Diagnostics, weighing the benefits against the risks, a little bit more on the side of the benefits in terms of an emergency use authorization, and looking at the risk, but there is always that balance that has to be looked at. And flexibility depends on the emergency situation. The benefits are ensuring that those devices and systems on the market are safe and effective, and I'll give you the definitions of safe and effective because there are some. And the risks are getting those to market as quickly as possible. And that applies to all of the IVDs, not just the emergency, but in the case of emergency, things get prioritized and pushed to the top. So I'll give you a general overview of FDA regulations of all in vitro diagnostics. Now, my area is infectious diseases, so anything, pathogens of any route were in the division that I headed up, but also within my office there's a chemistry division, there's an immunology division, there's a division of personalized medicine. But I'll be concentrating more on what I saw with the infectious disease area. But in general, the same regulations apply. And what I'm going to try and do is show you where there were differences and exceptions which are allowed to that general overview that we apply to every IVD under the emergency use authorization. So here's a definition of an in vitro diagnostic. You can see that it's 21 CFR 809.3. I won't quote too many of those regs, but it is in the regulation. And you can see it's the oversight of reagents, the instruments, and now we have to include software and the entire system that are intended for use in the diagnosis of a disease or other conditions, etc., 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 and also anything that's used to collect and prepare and examine specimens from the human body. And quite interesting now, over 70 to 80% of medical decisions are now based on some kind of diagnostic test result, together with, of course, the physician's overview, but they're used much more now in the diagnosis of diseases than they used to be. What's our authority? Federal Food, Drug, and Cosmetic Act. But it was only since 1976 that regulations were established for medical devices, which includes IVDs. Remember 1976, because when I think about what was on the market in terms of IVDs in 1976, wasn't much. There were radioimmunoassays, maybe, agglutination tests, gram stains, petri dishes, nothing much, okay? But you'll see that in one of the regulations that we have to follow, called the 510K, what people have to show is equivalence to anything that's been cleared since 1976. Think about what we have now on the market in terms of much more sensitive tests, and you can see how over time we've had to be very flexible in demonstrating that something was actually better than something that was approved in 1976. There's the Code of Federal Regulations that you get the IVD information from, and it includes the Quality System Regulation, Part 820. Now, as Cindy, I'm not sure she noticed that, but in fact, under the emergency use, we don't ask for any information about the manufacturing and quality system regulation, which we would do in normal circumstances. So there's one of the exceptions. FDA human subject protection regulations apply to in vitro diagnostics, the informed consent, the IRB review, and some of the things that apply to a clinical investigator that's running trials for IVDs. No informed consent needed for emergency use authorization, but as Cindy mentioned, there are the fact sheets, which are kind of substitute for that, letting the individual patient and the healthcare worker know some of the information about that particular test, and IRB review also isn't needed. One difference that we have in vitro diagnostics is that in our offices, in our divisions, we look at the whole product life cycle approach. You can see starting up at dividing it into post-market and pre-market. We like to get involved very early on, if there's a new technology, with the design of a particular in vitro diagnostic. So, as you heard from the drug and vaccine areas, we like to have pre-meetings with companies. It's actually free consulting. But one of the aims is to educate the reviewers in some new technology that maybe they were not familiar with. It also introduces us to a company it might be an entrepreneurial company, small company, who think that FDA is the dark side. And then once we meet with them, they usually say, oh, they're normal people. They're scientists like we are. So those kinds of pre-meetings, which never used to exist when I worked for industry. One of the main reasons I joined FDA was the promise that there was a new FDA coming up. That was 12 years ago, where we would interact with companies and behave like normal people and not policemen. So pre-market, pre-submission meetings, before anybody gets really into the analytical evaluations, help them with the design of the studies. And for the EUAs, for the in vitro diagnostics, in particular the molecular assays, only analytical evaluation was required. And for a normal non-EUA product, for the infectious diseases, pretty much all of them you need to have some kind of field clinical study. And the aim there is to see if the intended use of the product in the hands of the users is what the company is saying. And it's also, for us, ability to see if the package insert, the instructions, are easy enough for someone to use. We only asked for some clinical evaluation in certain circumstances, which I'll go into later with the Ebola products. If we go over to the post-market side or the compliance side, again, you can see that we did not ask for any information about manufacturing, which we usually do. We did, though, monitor both products that were being made in the US and exported out to West Africa without coming through FDA, and also products that were being advertised that also had not been through FDA. So there was a compliance issue there. There could have been export regulations applied for the products that were being manufactured here and exported, but those would have taken just as much time as going through our EUA process. But that was a piece of the FDA acting like a policeman and making sure that something that was made in the US and being exported was actually safe and effective. Post-market, that's surveillance to make sure the product is behaving the way that it was when it came through the FDA. That's very difficult during an emergency. We do have those as conditions saying, if you have adverse events, you need to let us know about it. But in fact, we know that under the conditions of an emergency, very often that's quite difficult for people to do. And in fact, we rarely, if ever, had anybody come back and say, it didn't work. What happens is we see a publication, perhaps, that's been done to say X product is not behaving the way that it's supposed to. That should be the company coming back and telling us that, but in fact, it doesn't happen during an emergency. During normal commercialization of a product, definitely the FDA holds companies to that because they have much more time. And then there are recalls of products. You know, usually with in vitro diagnostic, there are many lots made one after the other. Sometimes for no reason whatsoever, a lot will go bad on you. I've been there, done that. That's part of manufacturing in vitro diagnostics. And but as long as they tell the FDA or they have some recommendation of how to cure that product or that problem, then the product can either stay on the market or they have to recall it. So that's post-market type of information that we look at. Typical IVD device development pathway, again, recommending very early and often FDA interaction. You can see starts off with basic research, and then it goes on into analytical validation, which I mentioned is the important part of the data that comes in for emergency use authorization, clinical validation, yes, for normal product development and approval, data analysis, obvious for everything, submit and review. And never too early to seek FDA input. To any of you who are entrepreneurs, those are the people that we can really help, hold their hands. Why are we interested in that? Well, when we get a bad submission in, it's not fun at all. It's just not fun at all to have to go back and give them a 20-page deficiency letter of everything that they should have known if they'd have come in earlier. So it's good for the company to come in and find out what's needed. There are lots of tools. They can go into the FDA website and they can actually see the package inserts of different products that have gone before. They can see reviews of a product that maybe is similar to what you're trying to put through. And so there are many tools that can be used. There's really no excuse in this day and age for a very bad submission to come in. So works for the company, works also for FDA. Here are the definitions of safety and effectiveness. You can see that they are regulatory definitions. If you talk about safety and effectiveness, you see the scientific evidence is the core of this. And again, safety is reasonable assurances that probable benefits to health from use of the device outweigh probable risks, and that's where that balance comes in. And effectiveness is that reasonable assurance that the device in the target population will provide clinically significant results. Here are some of the pathways for in vitro diagnostics. Up on the left-hand corner there, you can see it's a risk-based regulation. In other words, if a product has false positives and false negatives, which, by the way, most in vitro diagnostics do, if I see a product that's 100% sensitive, 100% specific, I start to wonder, how did you get that data? All of these products have the potential to have false positives and false negatives for a multiple different reasons. So what we say is that for class 1 products, you don't really need very much information. We know a lot about that disease. For example, in vitro diagnostic infectious disease products, culture media, we don't need a submission for that. People just have to register and list it. If you put an antibiotic into that media, then it becomes a class 2, because now you actually make a decision about the any kind of issue when a bug has changed, and you get so you're using it for a different purpose whatsoever. You're trying to find out if the bug that's infecting that patient is actually resistant to that antibiotic. So that becomes a class 2. Most of the infectious disease tests are in class 2, which means they have some issues, and you have to have general controls. You have to have some clinical data, and all of the CBRN and the infectious diseases like Ebola would be class 2. So what's in class 3? Those are the most highly worrisome diseases where the effect of a false positive or false negative has some impact on the patient. Not very many. People always are scared off by that. For example, infectious diseases, HIV, hepatitis B, hepatitis C, papillomavirus, and a couple of the unusual TBs. We have downclassified hepatitis A and TB into class 2. The 510K de novo is a new kind of submission over the last 5 years, relatively new, where it used to be that any new product that came in where there was no previous test before that had been through FDA was classified as Class 3 and had to be put into a PMA. Big clinical trials, much more inspection involved with a PMA. Now when one comes in, we look at it and say, are there things, special controls that can mitigate those risks? And 100% of the time we have down-classified those into the Class 2 immediately. Very few IDEs for infectious disease IVDs. EUA, pre-EUA, I'll bring that up. Clear waiver. Do people know what clear waiver is all about? No? That's when you have a product like a rapid test, like a rapid strep test that can be used by someone who isn't a trained individual, that is not being run in a high complexity laboratory, in a hospital or in a reference lab, but can be used in a doctor's office or in the field. For example, those are the rapid tests that we'll be talking about later on. Those additional studies that you have to do to prove that they can be run in those kind of environments do take a little more time than the 510Ks or the PMAs. We realized up front that that wasn't something we could do in an emergency. You couldn't ask somebody with a rapid test to go out and spend another 3 months showing us that. So that was part of the 2013 recommendations. We were able to say, not required for EUAs. I'm not going to talk about LDTs, lab-developed tests, because that's another area of the tests that are out there, maybe being made in an academic institution that don't go through FDA because of something called enforcement discretion. There's an effort now to try and make some of those come into the FDA. Very controversial. Not going to touch that one. So, principles of a successful IVD submission to FDA, and this also applies to an EUA submission. What's the intended use? Is it clear and precise? Is there a complete device description? Is there scientific evidence supporting that intended use? And of course, we have to demonstrate safety and effectiveness, even if it is in an emergency. And the labeling, the package insert, etc., is really important. How much information do you need to demonstrate safety and effectiveness? Again, it goes back to the intended use, and I just mentioned that. What are the risks and consequences of a false result? And that drives that device classification and drives the amount of information the FDA likes to look at and review in a submission. Let's talk about the intended use. It's really the driving force of any scientific review, even including the emergency use or authorization type of devices. First thing, understanding the disease, conditions, integration of patient clinical management and public health surveillance. Yeah, we know that for influenza, but did we know that for Ebola? No, we didn't. I'll bring that up at another point, that this is one of the main things that people did not know enough about the disease at all, and didn't know enough about integration of the inpatient clinical management and public health in West Africa. Okay? Works for US, but not for West Africa. Who will be tested? Where and when? Did we know that about Ebola? No. What are the appropriate specimens? Didn't know that either. Timing, handling of them, how the results could be used in patient management. This all applies beautifully in the US, even for the emergency use authorizations. But as we saw, these things really were not known for Ebola. Device description in a pre-sub. This is what we like to see ahead of time. Tell us about what the principle of your assay is. What are the components and reagents, the instruments? What kind of controls do you have in there? How do you prepare the sample? Does it need some extensive extraction method like the PCRs? Preparation of the sample? What's the signal that's generated that you're checking? How do you interpret the results? And analytical testing results. I'll mention what is involved with analytical testing in a minute. And for if you're going to have a clinical, send us a clinical plan. What kind of sample size are we talking about? Is it statistically appropriate? And population, what's the availability of the samples? So let's look at analytical performance. What are we talking about there? So what you basically need to be able to say the performance of any assay is, what's the limit of detection? How low can you go to be able to pick that up? You can do all of those kinds of studies internally at your own site. You can do reliability. How reproducible is it if you use several different people running the assay? Just get the same answer. Is there anything that cross-reacts with this? Now, we know that the molecular assays don't have the same kind of issues as the serological assays. But you still have to look to see if are there any different viruses that might cross-react with that particular Ebola assay, let's put it that way. What kinds of things are typical in the population that you're trying to do testing in? In the US, obviously, different kinds of things in immunologically in the patient population than in Africa. I mean, we're not exposed to Lassa fever, et cetera, these were things that were very different about Ebola and had to be incorporated as we went along into some of the requirements for the cross-reactivity. Are there things that interfere with the product? For example, hemoglobin. Hemoglobin in serological tests can often cause false negatives. Rheumatoid factor in serological tests can cause false positives, not in a molecular test. There could be other things with molecular tests. Clinical performance. Some of it can be done internally, especially if you don't have access to specimens or actually in the field. We have a sort of level. Obviously, the best specimens that you can obviously use to demonstrate performance is fresh specimens directly in the field. Okay. Could we get those in Ebola? Not. Next best could be frozen specimens. Can you show there's no difference between a frozen specimen and a fresh specimen? You have to show us that. If you can use a frozen specimen. What if you can't even get frozen specimens? Spiking. Okay, you can spike into negative specimen, for example, virus. Next best. What about if you can't even get that for like H5 influenza? Then you have to come up with some sort of surrogate type of thing. With the molecular assays, you can do something even more. You can now do in situ, comparing with a library of DNA constructs. That's the kind of thing. We will allow all of that if somebody can show that they can't get the best specimen type. Labeling, very important. Again, you can see that in addition to intended use, we also put in there directions for use, warnings, limitations, result interpretation, as well as the performance characteristics. And then labeling for the EUAs are the 2 fact sheets. If you're interested in seeing some of the odds and end Q&A type things about in vitro diagnostic studies, like can you use foreign data, things like that, you can look this guidance document up. Emergency use authorization. So what lessons did we learn from the first time we used EUA in the H1N1 influenza pandemic? You heard about the EUA process. There can be 3 different determinations that there is an emergency coming from DoD, Department of Homeland Security, or HHS. But it's the HHS secretary who actually determines that declaration. But before that's done, there's a lot of high-level discussions from experts, from CDC, NIH, everybody, DoD, on do we really need to declare that emergency? Once it's declared, then we can start working on the EUAs, and if we issue an EUA, we're supposed to consult CDC and NIH. And again, when the emergency is over, all of those assays basically turn into pumpkins. They cannot be used again. You have to apply for a 510K or a PMA. That's something I keep telling people. As long as the Ebola crisis is there, as long as there's an emergency, these assays that have been authorized can be used. But once it's declared over, like H1N1, end of the use of those EUAs. You have to come through the proper pathway. I think you've seen most of this, so I'll cut this short, but basically we can get an emergency use authorization request from anyone, CDC, DoD, BARDA, commercial manufacturers, distributors, and again, it's not a substitute for getting the official approval, licensure, or clearance. It's not research, decisions made on a case-by-case basis, and again, we encourage submission of data prior to even a potential emergency in what's called a pre-EUA process, where we will look through all the data that people have and basically say, yes, if there's an emergency declared, you can get that out. We'll issue an EUA for it. But because the standard of issuance may be lower than the usual approval and clearance of an IVD, we've established conditions on the use. And some of those things and restrictions, I think, are kind of things that may come up this afternoon in the panel meeting. So authorized IVD devices between in 2009 until the end of the influenza pandemic. Just to give you an idea, the emergency was declared in April 24th. There weren't any cleared tests available that actually detected and differentiated influenza A subtype 2009 H1N1. There were rapid tests out there, but they had poor performance. The reason is obvious, because influenza changes so much that when those tests were cleared, maybe 10 years before, different strain of the virus, they were able to detect that one, but they were not able to detect the mutated virus H1N1. We received multiple EUA requests during this emergency from CDC, DoD. They were really, CDCs were to support the public health labs and only for use in those public health labs, DoD only for use in their laboratories for their personnel and families. Similar to the Ebola EUAs, CDC assays were only supposed to be used in the public health labs. One of the reasons is that they have no capacity for manufacturing surge capacity. And you'll see that in a moment. What happened in H1N1 is, yes, the first one was the CDC. Public health labs were overwhelmed with thousands and thousands of specimens. At first, the CDC did not want us to encourage commercial manufacturers, but those commercial manufacturers had that manufacturing capacity, had the availability of their instruments in multiple US laboratories. There's no point in having an emergency use authorized test where maybe they've sold it to 6 customers. Really, in an emergency, you need to have the instruments out there that can be used, and are in many laboratories. So just a quick overview of the summary of events for H1N1. At the top, you can see the emergency was declared on 4/26. We had reviewed over the weekend the data from the CDC. We'd actually seen some of it beforehand, and were able to give that an authorization the next day after the emergency. Why? I think I've explained why the first commercial assay didn't come for another 3 months. That was after everything got out of hand at the public health labs. But once we got that commercial assay out, we were able to, in the end, issue 18 different EUAs. Now, I want you to notice that they were all molecular assays, not rapid antigen assays. And over 1.5 million tests were sold in the US during that pandemic for the EUA. We developed some tools during that time. These were tools, we issued a document which explained what kind of information was needed. We established a standard template for the validation, and we found these to be very useful and things that we have continued to use during emergencies. We also found that a team approach and strong interactive review responses, instantly turning around, just like Cindy was saying, anything that came back in was really, really important. Also working with different agencies, we had a strong cooperation development with CDC at this time, their influenza group, and we also developed a very strong interaction with the DoD, which has continued to go on throughout. And of course, we streamlined the evaluation through these review templates. We had standardized review criteria, predefined standards recommended based on what we knew about flu. Did we have that for Ebola? No, not at all. Nobody knew very much at all about Ebola. There were some problems with limitation of clinical specimens, but again, we allowed a lot of flexibility even in the H1N1 area. And post-emergency, if you remember H1N1, suddenly there was a huge drop. It sort of disappeared, the virus. That left the companies with a gap because they couldn't get specimens to be able to get their 510K submissions. But the CDC and the public health labs supplied retrospective specimens to the developers so that they could get through and clear and approve their assays for general commercial use. We heard about the 2013 Preparedness Reauthorization Act. A couple of things that were important here was pre-positioning medical countermeasures. If somebody had a pre-EUA with us all ready to go, they couldn't put it in the laboratories ready to be, they had to keep them in their main area. That wasn't very good either. So for preparedness, if something had been pre-EUA'd, authorized, it could be placed in the laboratories ready to go. It also allowed the issuance of an EUA based on a potential for a public health emergency. That was new as well. And it permitted, as I said, basically for us to say, you don't need to do those clear waiver studies for rapid tests. So since then, 2013 and 15, we've had several public health emergency responses. We had influenza H7N9 developing in China, and a potential for an emergency was declared. The CDC came in with their assay as well as a commercial developer. If you notice, all molecular. 2 MERS-CoV EUAs have been authorized, both molecular, and 1 enterovirus D68 recently, a molecular assay. We have authorized FDA 10 Ebola Zaire assays, and there are 8 molecular and 2 antigen rapid tests. Ongoing issues, and I'll go into the Ebola much more, but for the others, still had the problem of needing characterized specimen panels. The unavailability of specimens for developers is apparent in all of the above. The cost of the test for molecular, obviously an issue. What are the incentives that the government can give to commercial developers to develop something for an emergency when there's no apparent market, and the need eventually to convert to a 510K? So quickly I'll go through some things with Ebola. It was way back in 2006 that actually the Department of Homeland Security said that the Ebola virus presented a material threat against the United States population sufficient to affect national security. But again, as you saw, it wasn't until August the 5th that the Secretary of HHS declared that circumstances exist justifying the authorization of emergency use of in vitro diagnostics for the detection of Ebola virus. Now I'll go through quickly, this is all public information, the different assays that have been authorized. The first, and if it says M after it, it means molecular. If it says AG after it, it says antigen. All this information you can find on the EUA website for the FDA. The first one that went through, actually, the day of the declaration, was the Department of Defense assay for Ebola Zaire. And why was it? Because we had seen it ahead of time. We had already authorized the pre-EUA submission 2 years before. The CDC were not ready. They did not have an assay ready to go. It took 2 months for the CDC assays, the next two you see, to come through on October the 10th. You can also see that there's been reissuance as people added specimen types, expanded their instruments, and so we've had several rounds of these. One thing that's come up with the DoD assay, now that the vaccines are out there, that their target is actually cross-reacts with the Ebola vaccines. So there's going to be some extra things done by the DoD to their assay. They're coming up positive in people who have been vaccinated with particular vaccines. None of the others have that problem. Down the bottom, the first one you see there, the BioFire, was actually a DoD contract with BioFire. BioFire is an instrument that has multiple assays that have been cleared on it. It's a multiplex type of assay, and it's very readily available. But this was a special one for DoD, and the next one below it is their commercial assay, which was authorized at the same time in October. Mention of the Altona. This is a German company. This was the first one that was authorized by WHO, again, a molecular assay. We authorized it in November based on the data that was sent in to us. It became the WHO's gold standard of which they used. We never declared a gold standard. NICE for us was the first major US company, Roche, which is the largest diagnostic company. That's the first time we were able to have a large company send in an emergency use authorization. You can see all the advantages I said about having a company that has surge capacity, has multiple instruments in different locations. But that was a first, and we were very happy that one of the big companies actually came in that knew a lot about how to submit things to us. Corgenics was the first rapid test, the antigen test. That was issued in February of this year. And Cepheid is an interesting molecular assay, which was given authorization in March because it's a self-contained a type of PCR. You get a result with that in an hour. Most of the PCR tests, you don't get a result for 4 to 6 hours. That includes the Altona. The BioFire, you get a result in 45 minutes. But these rapid antigen tests are 15 to 25 minutes. So big advantage of having a rapid antigen serological test. The Cepheid expert has the safety issue that's good because it's a self-contained and throwaway cartridge, so you don't have this issue with this very virulent virus. You can actually dispose of it. And the last one, which was just the antigen assay, the AuraSure antigen assay, was authorized at the end of July. That's it up to this point. So I want to just mention quickly some of the issues, and I know there are people in the audience who may have a different point of view, like to talk about that this afternoon, but these are some of the things where specific issues of serological tests, like an antigen test against a molecular assay. Developing a rapid serological test is much more complex and time-consuming than developing probes for a molecular assay. That's why you saw the molecular assays out very quickly. You can quickly develop probes for detection of the nucleic acid. Much more difficult to select and source the appropriate antigen-antibody pair to give the best test performance. Greater potential, maybe for false reactivity with related organisms. I mentioned that earlier, the rheumatoid factor, hemoglobin, etc., than molecular tests. And you cannot use in the studies in silico sources as a substitute for the cross-reacting organism studies. So that meant that delayed, in some cases, the emergence of an authorized rapid test. We didn't know much about the Ebola. Very few published literature, some by the CDC, saying that antigen detection was less sensitive in the acute stage of infection in the first 72 hours than the molecular tests. Lots of concern from lots of people about us authorizing and WHO authorizing an antigen test. Oh, there are going to be so many people that are going to be missed in that early stage. Well, I think we've seen some of the papers now that have been published to say that at the time people were tested, the viral load was very high and the antigen test was also possible. So again, perhaps this was really being very conservative at that point about antigen tests. But we had a lot of pushback from all sorts of experts about us even authorizing an antigen test. I think in retrospect, that was a lesson learned, that in fact those antigen tests are looking very good in the field. We did ask for some limited field study data for the first rapid antigen test under the field conditions, and we worked in parallel with WHO on getting that, because it was very difficult for anybody to get that kind of information in the field. They did spot a few things that they considered to be biosafety issues about the first test, about having something in a glass vial with rubber stoppers. There could be a problem of breaking the glass vial and breaking the barrier, etc., etc. So there were a lot of concerns about biosafety of using the antigen tests as well, which, again, with recent publications have been shown to be probably a little bit too conservative. But we used that data from the WHO field study to support the rapid test, the first one for EUA. Spend a few seconds on this because it's important to notice some of the things. You can look this up. You can look up the letters of authorizations on the website, some of the criteria that we put into the authorization letters. Again, it was emergency use for the presumptive detection of Ebola Zaire virus. Some people were concerned about that presumptive word. All it meant was really, if you have a chance to follow up on this later with we didn't call them confirmatory tests, you should probably do that because of some of the things. We did say that they should be used on individuals with signs and symptoms, not asymptomatic, had no information about asymptomatic individuals, and it should be used in geographic locations with high prevalence of Ebola infection. The more people there, the less false positives you get. So these are the kinds of things we put in there to try and mitigate some of the potential risks of not having a lot of data. There was no data to say that airport screening, any of these could be used in airport screening. A lot of fuss about that too, but you can imagine waiting 4 to 6 hours for a PCR or trying to, everybody that came in, do a rapid test. So to this point, nothing has been given that authorization. Again, based totally on scientific evidence and the benefits and risks outweighing the known potential risks, and that there was no adequate and approved alternative. Just for the antigen, though, we did have to put a few other little words in there. We did try and say, look, you can use this rapid test in situations where availability of a potentially more sensitive test is not available. And in fact, that's one of the scenarios in Africa where it was very difficult to get specimens from the patient to some of these mobile labs that were running PCR tests. Sometimes it would take days or weeks, which in this case was very detrimental. But we did say, look, okay, you can use the rapid tests in circumstances where you have nothing as sensitive as that. But you could also use them as well as the PCR tests as well. Other relevant sections of an authorization letter have some of those things in it. So I think this is my last slide, but some of the open issues that obviously have come up once we knew more about the pathology. Do need to authorize new sample types. We now know that even in convalescent patients, the virus can be present in semen. Because of the different funeral rituals, it would be important to be able to distinguish a cadaver of somebody who's died from Ebola than something else. We could get a claim for buccal cavity fluid. That would be the easiest way to be able to test to see if that individual had died of Ebola and had to be a different kind of burial than somebody who had died of something similar, but wasn't Ebola. Need for multiplex pathogen tests to rule out Ebola diagnosis. Unfortunately, Ebola has the same signs and symptoms as multiple other diseases in that area: malaria, Lassa fever, typhoid. And my division was able to get the recommendation from our lawyers that if there was a test like that that had Ebola in it plus others, it could be covered under the Ebola EUA. Need for tests to detect the other Ebola and hemorrhagic fever strains. And again, most importantly, need for rapid, less than 30 minutes, accurate, safe, easy to run, self-contained, perhaps molecular and antigen tests. And also, which held up many, many companies, the availability of samples, sample banks of well-characterized specimens for both molecular and antigen diagnostics. Very difficult. CDC had some ones they were only able to share with a few companies. I believe Tulane had some specimens that they were able to obtain. DOD had a few. But other than that, there was no sharing of specimens. Very difficult. We can talk about that later. So in summary, FDA regulates all IVD devices, including devices under EUA, and all based on risk assessment. The studies requested are to support the intended use claims. We ask for very few specimens. I can talk about that later for the emergency use claims. The challenges of medical countermeasures and these emerging viral pathogens have asked us to be very flexible in terms of working within the constraints of the regulatory law. And we did learn lessons from the 2009 H1N1 influenza pandemic. But obviously, we've learned a lot more lessons from working with Ebola. So information, you can get that. There's lots of information on our homepage. And I just wanted to give anybody who's a manufacturer the context now that I'm not the director. My deputy director, Uwe Schurf, is now the director. These are the branch chiefs in the Division of Microbiology that deal with the different kinds of diseases. And if you're interested in getting information about Ebola EUA, the senior reviewer contact now is Kim Sapsford. And what she will do, if anybody's interested, is we immediately send out to them what we call our templates. There are templates, draft templates. I have given copies to you, but you should get these from the Division of Microbiology Devices. Basically, it's fill in the gaps, and at the end of it, you have your EUA. And there's interactive review as you fill in the gaps all the way. This was one of the tools that we used throughout the Ebola. And one of them is for antigen, one is for molecular. So thank you.
Thank you. Thank you. I'm going to take a few questions.
Got time?
Yeah, let's just take, we have a break before the panel discussion. So let's take a few questions from Sally.
And I will go over a lot of these things that I mentioned. We have 10 minutes to talk about things in the panel meeting this afternoon too. Okay. So I'll bring up more things associated with Ebola. Okay. But if there are any questions in general, I'd be happy to.
Okay,
yes?
We'll take one. Sure.
You mentioned that there was a bottleneck in sharing of samples of specimens. Elaborate on that a little bit more. What kind of samples
I'll bring that up again later on, but you know, these are very pathogenic organisms requiring BSL-4 locations. Do you inactivate them? how does an inactivated virus work so you can ship those out? In many cases, the gamma radiation of different lots of the virus gave different results. So that was one of the first issues that came up. Only a very few individuals were able to, the CDC, Tulane, I believe, DOD, very few people had these viruses. And shipping these things around, unless you inactivated them, difficult. Getting specimens from West Africa, very difficult. A lot of international, the actual countries not willing to. That's been going on for a long time. H5, the Indonesians wouldn't ship anything out of Indonesia. MERS-CoV, same issues. People don't want to be considered to be the focal point of those pathogenic epidemics. It's very political. The whole thing was very political. In the end, working with BARDA and DOD and NIH, we began to put together panels that could be available. What stopped that? One of those things that, they were shipping out live anthrax, right, out of DOD. Immediately, everything stopped. So, many different issues. There were issues about, one of the things I would say to companies, can you get specimens from the patients, the US patients that came in? Wouldn't that be great? I mean, has anybody published, the serial bleeds or the serial, what happened during those things? Nobody's published them. Specimens not available. Companies had dibs on them, the people who were giving the vaccines and everything. Not available for diagnostic developers, etc., etc., etc. Okay.
We'll wait for the panel. You can see we have a lot of interesting things that we can discuss in the panel. Let's thank Sally and Cindy again. Thank you very much.